Okay, thank you, Lisa, and thank you, MVIF, for having me at this event. Let's go into presenter mode. Okay, I hope you can see my presented screen. So hi, everyone. My name is Mike Tisza, and I'm a newly minted assistant professor in the Department of Molecular Virology and Microbiology at Baylor College of Medicine in Houston, Texas, in the United States. Quick plug, since I will be opening my lab next summer, if anyone's interested in working in the wastewater virome or on other microbial genomics projects, feel free to reach out. You got my info here. My talk, I don't know how you think it fits, but I think for a field like microbiome sciences, so obsessed with human feces, we tend to think very little about what happens after we flush the toilet. And so I'm going to argue today that whole virome sequencing of municipal wastewater yields some really amazing biological insights, and it promises to have a real impact on public health. And I think there's a big connection because you can use some of the same microbial ecology tools and approaches that folks use for human gut microbiomes. All right, before I even get going, I want to do my acknowledgements first so I don't run out of time. I want to acknowledge the Texas Epidemic Public Health Institute, or TEPI, who supports this effort, and my colleagues there. And I especially want to acknowledge Dr. Sarah Yavernik-Krajine, who is the co-first author alongside me on a recent publication on this data. Okay, so there are many fundamental questions of virus epidemiology that we frankly do not understand at this point. Where do pathogens come from? That's something that's been talked about a lot. How are different populations affected by different viruses? How do non-biological things like climate, culture, and even the built environments of the cities we live in affect things like virus spread? And then also, and maybe most importantly, how do virus genomes evolve over time and place, and what affects those? What affects the evolution? Historically, we've tried to answer some of these questions with clinical and field data, which are great and they are invaluable. But the clinical and field methods are expensive and they can be biased. For example, the only people you're even going to see in the clinic are the most sick people. And so you're only going to get a biased snapshot. On the other hand, I think wastewater presents a really unique opportunity as everyone, that's me and you and everyone here listening, contributes to the wastewater, right? Every time you flush a toilet, every time you wash something down the drain. So in that way, it's relatively unbiased. And you can anonymously sample a whole town or a whole community with a single sample, and you can do this all the time, and you can see how things change. The next component of this is viruses. Viruses are very tricky. Within the virosphere, there's actually much, much more diversity than there are in cellular genomes. Even though we know how diverse and unique and kind of crazy bacteria are, viruses are many times more diverse. So what we do is we find the common denominator between all viruses, and that's nucleic acid, right? And so all human and animal viruses encode their genome with nucleic acid in DNA or RNA. And so we can use that to pull them out, and I'll explain how that works really quick. So our whole program with this wastewater sequencing uses the following approach. So basically, we extract DNA and RNA from wastewater influent samples that we get from treatment plants. We synthesize cDNA from the RNA, so it all becomes DNA. We then prepare barcoded libraries as you would for sequencing, and then we put these barcoded libraries on a huge library of biotinylated oligoprobes. And these probes are 120 nucleotides long, and they are synthesized based on the genomes of about 3,000 known human and animal viruses. When you dump your wastewater libraries on there, the fragments from virus genomes bind readily to these biotinylated probes because it's sequence specific, and everything else, or most of the other stuff, gets washed away. And then you pull down your virus fragments, you sequence those, and then you do some bioinformatic analysis to find out, so you can detect, you can quantify, and you can call the variants of all the viruses in your sample. For our purposes, I use a software I wrote called S03 to do this. Okay, so what do we get? Well, first I want to show and emphasize how powerful this method is. So if we took, here showing the yellow on the left, if we just took sequencing libraries from wastewater and we didn't do the hybrid probe capture that I just explained, we might get 2 or 3 reads per million reads that map to any human or animal virus. Not very much, but when we do enrichment, we get over a 3,000-fold enrichment of virus reads, typically giving us hundreds of thousands of virus-specific reads in a 20 million read library. So we can do a lot with that many reads that map to viruses. And in fact, if you add it up over across all the samples we've done so far, we actually detect different viruses, hundreds of species from 28 different virus families, and that includes all Baltimore classifications of viruses, right? Single-stranded, double-stranded, DNA, RNA, reverse transcribing. We can detect them all, and we do. And so a little more about our program, just to talk about the scope of it. We take weekly samples now from 21 different wastewater treatment plants across 9 different cities in the great state of Texas. And on the right here, it shows a chart of all the samples we've collected. And some places, like in Houston and El Paso, we've collected over 18 months of data. And so for the sake of this presentation, I'll now just be focusing on Houston and El Paso because we have the most data from those cities, and we sample at the most number of unique sites in those cities also. All right, so if this method is going to be useful for public health, it should give you the virus levels that reflect the community disease burden, right? It should be basically quantitative. So if you look at the charts here, if you start, we'll start looking at this one on the right here for influenza A virus in Houston last year. I hope you can appreciate that the virome-based sequencing, which is this gray dotted line, closely follows the influenza positive hospital patients in Houston, which is this purple line. So basically what we see is that the wastewater values for influenza go up and down when the clinical burden goes up and down. And then on the left, you can see something similar, but this is virome sequencing, again, with these gray dotted lines for SARS-CoV-2 levels in Houston and El Paso, respectively. And then these yellow lines are the positive clinical PCR tests in Houston and El Paso, respectively. You might be less convinced by this data because the lines don't follow each other as well. But this time period, last summer and winter, corresponded with the time when people were basically abandoning clinical PCR testing in favor of rapid antigen tests. So that's why the number of tests didn't rise so much on that second wave. And furthermore, our methods with virome sequencing correspond to levels in wastewater based on other methods like qPCR. So we know it is quantitative. Some more clinical correlations if you weren't convinced already. So on the left, we were able to pick up last year's monkeypox outbreak in Houston. Even though there wasn't so many cases, this method can be very sensitive. And so we can see it here. Again, this gray dotted line is the virome sequencing. And then another virus that correlated very closely with the available clinical data was enterovirus D68. Again, the gray line is the virome sequencing and the yellow line is the clinical data. Okay, so, but we can do more than just track these really nasty pathogens. We can actually do whole community analyses. We can do all the alpha diversity, beta diversity, all this kind of stuff. And so I wanted to show that. So basically this is the microbial ecology part. So each dot here represents a single wastewater sample. Its color corresponds to its city of origin, either Houston or El Paso. And this dimensionality reduction plot puts dots that have similar community compositions near each other, basically, right? So if the dots are close, the virus communities are similar. And I think what is strikingly obvious is that Houston and El Paso each have their own unique viromes that don't overlap too much with each other. I was really excited to see that. But the other thing I want to show too is that time matters. I'm just going to let this play a couple times. But this is community data from 4 wastewater treatment plants in El Paso. And you can see how this virome changes over the year. As you see, the dates change here, and it goes literally full circle And you see that time is kind of a flat circle here, so that the virome communities change a lot, and then they go right back to where they started, and then they start to change again. Okay, so I'm running out of time, so I will say here we can also look at mutational profiles of certain viruses and see how they're evolving. So and this does depend on the amount of viruses in the wastewater, and we need to get enough coverage and all that. But here we're looking at human astrovirus MOB1. And you can see the mutational data, the non-synonymous mutations plotted across different cities. The bottom being El Paso, the top being Houston, and then the y-axis being dates. And so I try to highlight a couple areas of the genome here where you see differences. For example, for lack of a better term, this pink smudge here, you see it in El Paso, but you almost never see this variant in Houston. And similar, Houston has a variant here that you don't really see in El Paso. And you have similar things going on in the capsid gene. And so that is the gist of what I wanted to talk about today. Comprehensive virome sequencing of wastewater is facilitated by probe-based hybrid capture methods. It reliably tracks virus pathogens in the communities, and it reveals spatiotemporal patterns of the virome community and virus evolution. Thank you.